Hello everyone. In this video I'm going to talk about the mechanism of action and synthesis of Adagrasib, a pharmaceutical approved by the FDA in 2022 for the treatment of non-small cell lung cancer. KRAS, Kirsten-Wright sarcoma virus, is a gene located at chromosome 12 in humans that provides instructions for making a protein called KRAS. The protein is a relatively small membrane-bound GTPase that relays signals from outside the cell to the cell's nucleus. In a previous video, I talked about the mechanism of action of sotorasib. Basically, in normal cells, KRAS is activated by binding to GTP. This promotes the activation of multiple downstream signals that regulate cell survival, growth, and proliferation. When GTP is hydrolyzed to GDP, KRAS is inactivated. The KRAS gene is frequently mutated, and a frequent mutation involves the substitution of glycine 12 by cysteine. Such codon 12 mutations disable GTP hydrolysis by GTPase activating proteins, GAPs, and maintain KRAS in its active form, leading to uncontrolled cellular proliferation and growth. Adagrasib, like sotorasib, is a covalent inhibitor of KRAS G12C that suppresses KRAS G12C signaling and tumor cell growth by binding to the GDP-bound form of the protein. The model suggests that the RAS inhibitor and guanosine exchange factors, GEFs, compete for binding to KRAS GDP. Drug-bound KRAS GDP is not susceptible to exchange factors and exits the cycle, thus lowering the levels of active KRAS. RAS protein is a featureless, nearly spherical structure with no obvious binding sites, making it difficult to synthesize compounds that can effectively target and inhibit its activity. A structural biology analysis has identified two switches, switch 1 and switch 2, on the surface of the KRAS protein that change their status based on the binding status of KRAS. The switch 2 region provides an entry point for allosteric regulation. Allosteric inhibitors that interact with mutant amino acids in switch 2 region can induce conformational changes, resulting in an inactive KRAS conformation. The KRAS G12C mutant protein contains a mutant cysteine, cysteine 12, which provides a potential covalent site for inhibitors. When an inhibitor with a covalent warhead binds covalently to the mutant cysteine 12, it induces a new allosteric pocket, the switch 2 pocket, in the switch 2 region. The small molecule inhibitor then interacts with the corresponding amino acid, resulting in a conformational change of the KRAS protein. These effects reduce the affinity between GTP and KRAS, preventing GDP from being replaced by GTP through GEF catalysis, and ultimately lock KRAS mutants in an inactive state. Sotorasib and adagrasib achieve inhibitory effects by stabilizing the conformation of KRAS G12C mutants in an inactive state through covalent binding to residues in the allosteric site. This highlights the significance of covalent inhibitors and allosteric regulation in drug discovery, particularly in targeting KRAS-related diseases. Adagrasib features three distinct subunits, an N-methylprolinol, a chloranaftyl, and a substituted piperazine attached to the tetrahydropyridopyrimidine core. The unique 2-fluoroacrylamide warhead located at the distal site of the piperazine is responsible for covalent binding to the target protein. The current commercial synthetic route starts with the Boc-protected tetrahydropyridopyrimidine. A regioselective SNAR reaction introduces the CBZ-masked piperazine moiety at the more reactive 4 position. In general terms, nucleophilic aromatic substitution reactions on pyrimidines proceed faster at the 4-6 position than the 2 position, but it also depends on the substituents and the reaction conditions employed. Installation of the prolinol subunit is achieved via palladium-catalyzed carbon-oxygen bond formation between the pyrimidine at C2 position and the prolinol. The desired product is then isolated as the tartaric acid salt. Introduction of the chloranaftyl subunit is achieved with the free base, followed by a Backwell-Harbig elimination with chloranaftyl bromide. Practical conditions employing 2-mercaptoethanol were developed to unmask the CBZ-protected piperazine to afford the desired compound. Trimethylsilyl iodide is a traditional deprotection reagent for this transformation. However, benzyl iodide, which is a potentially mutagenic impurity, was coproduced. Elimination of this impurity was difficult to remove because it could be trapped by the basic site, the tertiary amine of the prolinol, generating a quaternary ammonium species. The conditions employed cleanly release the benzyl group and carbon dioxide. The coproduct of the reaction, thioether, easily purges, and the thiol precursor is surprisingly minimally odoriferous. Propanphosphonic acid anhydride, T3P-mediated coupling of the piperazine with sodium 2-fluoroacrylate completed the synthesis of Adagrasib. Thank you for watching. I hope you enjoyed the video. Please subscribe, like, or leave me a comment down below. I'll see you in the next video. Bye!